Imagine a small house on a quiet street in Hartford, Connecticut, in the earliest years of the 20th century. The windows are thrown open to let in the soft breath of June, and the shadows of elm leaves tremble across the wooden floor. Inside, a little girl sits alone on a worn carpet, arranging a small collection of pebbles and dried flower petals she gathered that morning. Her name is Eleanor, but everyone calls her Barbara. She is 4 years old, and already she carries a stillness within her that makes adults pause. The light falls in warm, dusty beams across her lap as she sorts the tiny treasures with careful, deliberate fingers. Her parents, a physician father and a piano-playing mother, move gently through the rooms behind her, their voices low and unhurried. This is a household where solitude is respected, where a child's curiosity is not hurried along, but allowed to unfurl like the ferns in the garden outside. From her earliest days, Barbara McClintock inhabited a world shaped more by the rhythm of growing things than by the noise of people. The family would soon move to Brooklyn, but the pattern was already set. She was a shy, self-contained child who found comfort in what the earth offered up: stones, insects, the way light slanted through a leaf. In the backyard, she would crouch for an hour to watch the slow progress of a beetle across the dirt, her shadow pooling around her like a dark puddle. No one interrupted her. Her mother understood this quiet absorption. The neighborhood children played hopscotch and jumped rope, their laughter ringing down the block, but Barbara often stood at the edge of the game, a faint smile on her face, her mind already walking along a different path. She felt no loneliness, only the gentle pull of questions no one else seemed to be asking. When the family moved to a semi-rural area of Brooklyn, the landscape opened up to larger patches of wild grass and vacant lots where goldenrod and Queen Anne's lace swayed in the breeze. There, Barbara discovered the deep pleasure of being alone with growing plants and the humming communities of insects that lived among them. She would lie flat on her stomach in the warm grass, her chin resting on her folded arms, watching ants trace highways through the green. The hum of a distant lawnmower, the occasional bark of a dog, the scent of clover, all these impressions settled into her like stones sinking into clear water. At dusk, the sky took on shades of lavender and honey, and the streetlights flickered on one by one, spilling small pools of amber onto the pavement. Her mother's call for supper would float out across the yard, and Barbara would rise slowly, reluctant to leave the deepening twilight and the tiny, unseen worlds that continued on without her. Though she felt a strong connection to her father, Dr. Thomas McClintock, it was her aunt who gave her the first instrument of formal observation, a small magnifying glass, wrapped in brown paper. Barbara held it up to a blade of grass and gasped softly at the architecture revealed there. The parallel veins, the tiny hairs, the cells stacked neatly like bricks in an infinite wall. That moment shifted something inside her. She began to see the ordinary world as a place of hidden structure, a vast, silent city of patterns waiting to be seen. The magnifying glass became her constant companion on long, meandering walks to the edge of town. She would kneel beside a puddle after rain and study the way the water reflected clouds, or trace the delicate skeleton of a fallen leaf until the sunlight began to fail. This was not mere child's play, it was the first murmur of a vocation. At home, the evenings were filled with the sound of her mother's piano drifting through the downstairs rooms, soft as a fall of leaves. Barbara would sit at the window seat, a book in her lap, but her eyes were often lifted to the evening sky, watching the slow crawl of shadows across the lawn. Her father, returning from his rounds, would pause in the doorway and simply stand there, observing his quiet daughter with a gentle curiosity of his own. He never asked her to be more talkative, more sociable. He seemed to understand that her mind needed room to drift. The family ate dinner by candlelight sometimes when the electric light felt too harsh, the flames dipping and bowing as someone reached for bread or passed the salt. In that soft, wavering light, Barbara felt the day's outdoor brightness distilled into a warm interior glow, and she would carry that feeling upstairs to bed, her small magnifying glass resting on the nightstand among smooth stones and a feather or two. School, when it came, proved to be an uneasy fit. The classrooms were loud and full of rigid routines, and Barbara found herself retreating into daydreams that were far more interesting than the lessons. She would gaze out the window at the schoolyard tree, watching the way the wind turned each leaf into a small, fluttering hand. The teachers often reprimanded her for not paying attention, but what they could not see was that she was paying a different kind of attention to the shifting patterns of light, to the slow unfolding of spring buds, to the hidden logic of the natural world. Her mother was called in for meetings, and she would listen politely, but back at home she never scolded. She recognized something of herself in this solitary child. Some evenings, after supper, Barbara would go into the garden and dig gently in the cool earth, feeling the damp soil under her fingernails, and all the noise of the day would wash away. By the time Barbara reached high school, the family had settled into a comfortable rhythm that left plenty of space for her quiet pursuits. She discovered the high school science room, a long chamber filled with wooden cabinets, glass specimen jars, and the faint smell of formaldehyde, and it felt like coming home. Her science teacher, a soft-spoken man with spectacles that always sat slightly askew, noticed her at once. He saw how she handled the microscope with a reverence that other students lacked, how she adjusted the focus so slowly, so patiently, as if she were coaxing a shy animal from its hiding place. After school, when the corridors emptied and the last light of afternoon slanted through the tall windows, Barbara would stay behind to examine slides of onion root tips and pond water, her breath quiet, her world narrowed to a perfect circle of light. In those moments, the seed that had been planted in the grass of her childhood began to send up its first green shoot. The path from that quiet high school laboratory to Cornell University in 1919 was as natural as a stream finding its course downhill. Barbara arrived on the Ithaca campus during the golden flush of autumn, when the hillsides around Cayuga Lake were on fire with orange and crimson. She carried a single suitcase and a heart full of unspoken determination. The campus itself seemed like a vast, orderly garden, stone buildings covered in ivy, winding paths lined with elm trees, the distant clang of a bell tower marking time without hurry. She found her room in a women's dormitory, a small, plain space with a window facing east, where the morning sun would wake her gently. That first night, she sat on the edge of her bed and listened to the unfamiliar sounds of the building settling, of other young women laughing softly in the hall. She felt, for the first time, the edges of her solitude expanding into something like freedom. At the College of Agriculture, Barbara enrolled in plant breeding and cytology courses that would shape the entire architecture of her life. The lecture halls were grand, with high ceilings and blackboards that stretched wall to wall, but her true domain was the laboratory, a long room on an upper floor where the windows ran down one whole side. In the late afternoon, the light would pour through those windows in thick, honeyed sheets, illuminating rows of brass microscopes that stood like patient sentinels. She would sit at her bench for hours, her posture perfectly still, her eye pressed to the eyepiece. What she saw there in the cells of corn plants was a universe no less vast than the starry sky. Chromosomes, stained a deep ruby red or purple, lay tangled and elegant within the nucleus, and as she watched them divide, she felt the same quiet awe she had known as a child with her magnifying glass, only infinitely magnified. The early 1920s at Cornell were a time of extraordinary ferment in genetics, though the word itself was still young. Barbara's professors recognized her unusual gifts immediately. She had a patience that others lacked, an ability to sit with a single specimen for an entire afternoon, adjusting the light, making minute notes in her neat script. Outside the lab windows, the seasons wheeled by, the first snow dusting the quad, the sudden green explosion of spring, the heavy heat of summer when the cornfields rustled like dry paper. Barbara moved through these seasons as if they were the backdrop to a deeper calendar, an interior rhythm governed by the life cycles of the plants she studied. She learned to prepare her own slides with exquisite care, fixing cells onto glass with a delicacy that came from years of handling fragile things. Her classmates sometimes watched her in respectful silence, understanding that something rare was unfolding there in the quiet corner by the window. It was during these years that Barbara developed the technique that would make her name among cytologists, a method for staining and spreading maize chromosomes so clearly that they could be mapped like roads on a cartographer's chart. This labor required steady hands and an almost meditative concentration. She would rise before dawn and walk down the empty, dew-soaked paths to the laboratory, where the air still held the chill of night. Alone, she would light the Bunsen burner, its soft blue flame wavering then steadying, and begin the long process of teasing apart the tiny strands of genetic material. The world outside was silent but for the occasional bird call. As the sun finally rose and the room filled with pale gold light, she would step back, stretch her shoulders, and look down at the slide. Under the microscope, the chromosomes appeared like tiny X's, neatly arranged, each one telling a story that no one else had yet learned to read. Her doctoral research focused on the chromosomes of maize, an organism that became, for Barbara, a companion as much as a subject. She grew corn in the experimental plots behind the greenhouses, spending long mornings walking between the rows, the tall stalks rustling above her head, their tassels tossing pollen into the warm air. She wore a simple cotton dress and a broad-brimmed hat against the sun, and her hands grew calloused from the work of cross-pollination. The corn plot was her outdoor laboratory, a living library of variation. She would kneel in the rich, dark soil and examine the developing ears, noting the color of each kernel with the same precision she applied to chromosomes. A red kernel here, a white one there, a pattern of spots, each told a story of inheritance, of crossing over, of something deeper still that she could not yet name, but could feel like a faint vibration beneath the surface of things. By the time Barbara earned her PhD in 1927, she had already secured a place within the small, intensely collaborative community of maize geneticists. She stayed at Cornell as an instructor and researcher, her daily rhythm unchanged. She lived alone in a small, rented room near the campus, its single window framing a silver birch tree whose leaves chattered softly in the breeze. Her meals were simple: bread, cheese, fruit taken at a wooden table while she read the latest journals. In the evenings, she would walk down to the lake and sit on a weathered bench, watching the water darken from blue to deep indigo. The gorges around Ithaca echoed with the sound of waterfalls, a constant, soothing backdrop of white noise. She rarely felt lonely. The company of her thoughts was rich, and the maize plants seemed to whisper their secrets to her in the rustling of their leaves. She was, in the truest sense, a scientist made for the long, slow observation of nature. The next chapter of her life was unfolding even as she sat by the lake. She had begun to notice odd, unsettling patterns in the inheritance of kernel colors that could not be explained by the standard model of genes fixed in place on the chromosome like beads on a string. Something was shifting, something was moving. She had no dramatic eureka moment, no flash of lightning, no shout of discovery. Instead, the understanding came as softly as the dusk settling over Cayuga Lake, a slow recognition that the genetic material was more fluid, more alive, than anyone had dreamed. She would carry this glimmer with her through the next decades, through solitude and indifference, like a small, steady flame cupped in her careful hands. The Great Depression had tightened its grip on the nation by the early 1930s, but Barbara McClintock's work continued with a quiet tenacity that defied the economic gloom. In 1931, she published a landmark paper with colleague Harriet Creighton that provided the first direct evidence of genetic crossing over, a phenomenon long theorized but never physically seen. The news caused a ripple in the scientific world, and Barbara's reputation as a first-rate cytologist was secured. Yet for all that, permanent academic positions for women were vanishingly rare, and she found herself moving from one temporary post to another, her suitcase perpetually half-packed. She spent time at the University of Missouri, at Caltech under the warm Pasadena sun, always welcomed, always respected, but never quite rooted. Through all the shifting, she carried her meticulously prepared slides in a wooden box, cushioned with cotton, as if they were priceless relics, and to her, they were. Each slide held a piece of the puzzle she was slowly assembling, a map of a territory no one else was exploring. In 1941, she received an invitation that would become the geography of the rest of her life. The Carnegie Institution of Washington offered her a research position at the Cold Spring Harbor Laboratory on the north shore of Long Island. She arrived in the winter, when the bare trees stood like dark lace against a pale sky, and the harbor itself was a sheet of quiet gray. The laboratory was a cluster of modest buildings set among rolling lawns and old oaks, with a view of the water that changed with every hour. Her new workplace was a small, white-clapboard building that housed the genetics department, its rooms lined with wooden benches and the familiar smell of fixatives and stains. She was given a plot of land behind the lab where she could grow her corn, and she immediately began to prepare the soil, her hands remembering the feel of the earth. That first spring, as the green shoots broke through, she felt a deep sense of arrival. This was to be her home, her sanctuary, for more than half a century. Day after day, season after season, Barbara tended her cornfields and her microscope with the patience of a monk. The Cold Spring Harbor summers were lush and green, the air thick with the scent of salt water and growing things. Mornings, she would walk down to the field before breakfast, the dew soaking the hem of her dress, and check each plant for subtle changes in leaf color, in growth pattern. She knew every stalk as a shepherd knows her flock. In the laboratory, she began to observe something deeply puzzling in the way certain patterns of pigmentation in the kernels were inherited. A kernel might be colorless, then spotty in the next generation, then back to deep purple, as if some controlling element had been switched off and on, moved around. The existing theory said genes sat in fixed locations on chromosomes, passed down unshuffled except for rare crossing over. But what Barbara was seeing suggested something far more dynamic, genes that could, in effect, jump from one place to another. She named these mysterious entities controlling elements and spent years meticulously tracking their behavior. Her tools were simple: a sharp eye, a camel's hair paintbrush for cross-pollination, and an unshakable conviction that the maze would reveal its secrets if she only watched long enough. The painstaking work required her to grow thousands of plants, hand-pollinate each one, then examine the individual kernels under a dissecting microscope, counting color patterns like a census taker counting households. She filled notebook after notebook with neat columns of data, sitting in her small office as the afternoon light turned the paper golden. Outside, the world was at war, but inside the quiet lab, the only tension was between the accepted doctrine of stable genes and the revolutionary hints of movement that were emerging from the cells. Barbara felt no rush to publish. She was, as she later said, just trying to figure out what the corn was trying to tell her. The great revelation came together slowly over many years, not as a sudden burst, but as a picture gradually clarified. Barbara realized that the genome was not a static string of instructions, but a living, responsive system. Transposable elements, later to be called transposons or jumping genes, could detach from one site on a chromosome and insert themselves into another, altering the expression of neighboring genes. This was a direct challenge to the orthodox view that the gene was a fixed, unchanging unit of heredity. The implications were staggering, but Barbara's primary emotion was not triumph, but a quiet, profound satisfaction. She had deciphered a language written in the stains and bands of chromosomes, a language of movement and change. In the solitude of her lab, she would lean back from her microscope and gaze out the window at the cornfields bending in the wind, and she felt a kinship with those plants that went beyond science into something like reverence. In 1951, Barbara presented her findings at the Cold Spring Harbor Symposium, a gathering of the most prominent geneticists of the day. She stood at the podium, a slender woman in a simple dress, and laid out her evidence with unassuming clarity. The room was filled with brilliant minds, and the air was heavy with the smoke of cigarettes and the weight of expectation. As she spoke, describing the movement of these controlling elements, a hush fell over the audience. But it was not the hush of amazement, it was the silence of incomprehension. When she finished, there were few questions, no applause. The prevailing belief was that genes were stable beads on a string, and what Barbara was proposing seemed almost mystical, a step too far. She left the symposium feeling that she had spoken a language foreign to everyone who heard it. Walking back to her small cottage on the laboratory grounds, the night air cool on her face, she felt the first heavy wave of professional isolation. The years that followed were difficult in a quiet, interior way that never made headlines. Barbara continued her research, publishing papers that were largely ignored or dismissed as too complicated. She withdrew somewhat from the larger scientific community, choosing instead the company of her corn and a small circle of trusted colleagues. The non-reception of her greatest discovery became a kind of open secret. Invitations to speak at major conferences dwindled. Yet, strangely, she did not seem embittered. Neighbors saw her in her garden, weeding or planting, her movements deliberate and calm. She gave a friendly wave but did not stop to chat. The cornfields grew taller each summer, their tassels shaking out clouds of pollen onto the bright air. And inside the small white building, the work went on, the patient accumulation of proof, a quiet monologue addressed to a future that had not yet learned to listen. The 1950s deepened into the 1960s, and Barbara McClintock continued her solitary rhythm of life at Cold Spring Harbor. The world outside was changing rapidly. Television antennas sprouted on rooftops, jets cut white trails across the sky, and molecular biology was racing forward on the strength of the double helix model. But inside her modest cottage, time seemed to move at a different pace. The cottage was a simple two-story structure of pale shingles, set among huge old oak trees that cast dappled shade through every season. She awoke each morning to the sound of birds in the branches outside her window and the distant lapping of the harbor tide. Her breakfast was a piece of dry toast and a cup of strong tea that she carried to the porch, where she would sit in an old wicker chair and watch the light change over the water. These minutes were as essential to her as any laboratory equipment, a quiet calibration of the inner self before the day's careful looking began. Her daily walk to the laboratory was a ritual unchanging in its details. She followed a gravel path that wound past a small pond where mallards drifted lazily, their reflections trembling on the surface. The path was bordered by wild roses in summer and by stands of goldenrod in the fall, and she would often pause to examine a particular flower or insect, her scientist's eye never at rest, but softened by a gardener's affection. Upon reaching the lab, she exchanged a few quiet words with the custodian, a gentle man who kept the floors polished and the windows clean, and then she climbed the stairs to her small corner room. The room was a haven of piled journals, cardboard boxes of dried corn samples, and the steady, warm glow of a desk lamp. There was no telephone, no radio, only the whisper of the heating system and the occasional tap of a branch against the window glass. This was the environment she had chosen, deliberate, insulated from the noise of disbelief, a chamber where her private conversation with the maze could continue uninterrupted. Through all those years, her research moved forward, though only a handful of people paid any attention. She continued to grow her corn, to hand-pollinate each plant, to sit at her microscope for hours that melted into days. She mapped with increasing precision the movements of the transposable elements, and she even developed a sophisticated theory about how they might regulate the expression of other genes, acting as a kind of genetic control panel. In her notebooks, the data grew into an intricate tapestry of evidence so dense that few other scientists had the patience to follow its threads. Yet, she never doubted the validity of what she was seeing. The corn was her faithful informant, never lying, never rushing. She would often say, in later years, that the key was to let the organism tell you its story, to be quiet and listen. And in the deep stillness of those decades, she listened harder than anyone. The changing of the seasons became the primary punctuation in her life's sentence. Autumn at Cold Spring Harbor was a blaze of russet and gold that seemed to set the whole landscape alight. Barbara would walk through the falling leaves in the late afternoon, collecting stray ears of corn that had been missed in the harvest, her breath forming small clouds in the crisp air. Winter brought a profound silence, the snow blanketing the fields and muffling all sound, and she would work in the lab under the yellow electric light, her mind as clear and sharp as the icicles hanging from the eaves. Spring was a slow, muddy stirring, and she would be out in the field as soon as the ground thawed, her boots squelching in the wet earth, turning the soil with a sense of renewal that never grew old. Summer, with its heavy, humid days and the constant thrum of cicadas, found her in the shade of the cornstalks, measuring, noting, being. Her solitude was not absolute. She maintained a steady correspondence with a few trusted colleagues, writing long, thoughtful letters in her precise hand. And she occasionally traveled to give lectures, though she was aware that many in the audience came to see the eccentric lady who talked to corn rather than to absorb her science. She bore this with a quiet dignity, her presentations as carefully organized as her gardens, her voice calm and even. Back at Cold Spring Harbor, there were the yearly summer symposia, which she attended as an unobtrusive presence, listening politely to the young researchers who were mapping the new world of molecular biology. Sometimes, a perceptive student would approach her, drawn by some rumor of her work or simply by the hint of deep knowledge in her kind, watchful eyes. She would then guide them gently through her data, speaking not as a defender of a forgotten theory, but as a storyteller recounting the history of a far-off, intricately beautiful land. As the 1960s drew to a close, the first small cracks appeared in the wall of silence that surrounded her. Other scientists working with bacteria and viruses began to notice odd, movable pieces of DNA that eerily resembled the controlling elements she had described in maze nearly 2 decades earlier. The language of molecular biology was beginning to converge with the cytological language she had pioneered. There was no dramatic phone call, no sudden public vindication. Rather, it was a slow, dawning light, a shift in the collective awareness of the field, as young researchers came across her old papers and realized, with a shock of recognition, that she had already been where they were now arriving. Barbara, for her part, received this quiet validation with the same equanimity with which she had received the silence. She had never needed the world's applause. The work itself was the meaning. And in her garden, the corn continued to grow, tall and green, as if it too understood that patience is the deepest form of trust. The slow tide of recognition began to rise more steadily throughout the 1970s. As molecular biologists delved deeper into the mechanisms of gene regulation, they kept bumping into the same phenomenon, pieces of DNA that moved, that switched genes on and off, that did not obey the static theories of the old textbooks. Barbara McClintock's name began to appear with increasing frequency in footnotes, then in article after article, as researchers realized they were not discovering something new but confirming something long known. She received the first major public honors of her career, the National Medal of Science in 1970, the Albert Lasker Award in 1981, and several honorary doctorates from universities that had once politely ignored her. At each ceremony, she stood quietly, her silver hair neatly combed, her voice soft but clear, receiving the plaudits not with false humility but with a calm awareness that these moments were the world catching up, not her own arrival. The spotlight was warm, but it shone on a woman who had long ago made her peace with the shadows. It was a cold October morning in 1983 when the telephone rang in Barbara's small cottage at Cold Spring Harbor. She was, as usual, preparing her simple breakfast, the kitchen filled with the scent of toasting bread. The caller informed her that she had been awarded the Nobel Prize in Physiology or Medicine, unshared, for her discovery of mobile genetic elements. She was 81 years old. When reporters later asked how she felt, she replied with characteristic simplicity that she was pleased, but that she had known for a long time that the work was correct. There was no great astonishment, no burst of emotion. It was, she said, like being given a lovely dessert after a very long and satisfying meal. The real nourishment had come from the years of quiet observation, the countless hours at the microscope, the conversations with the corn. The prize was a grace note, not the symphony. In the days that followed the announcement, the quiet pathways of Cold Spring Harbor were suddenly filled with journalists and television crews, their bright lights intrusive against the gentle, faded landscape. Barbara handled them with a blend of patience and bemusement, giving short, thoughtful interviews that seemed to puzzle reporters looking for sensational sound bites. She would often lead them out to the cornfield, still growing in the same small plot behind the lab, and gesture gently at the dried stalks bowed under the autumn sky. "Here," she would say, "this is where the thinking was done." The camera's word, capturing an elderly woman in a practical coat, her hands buried in the pockets, her eyes scanning the brown tassels as if still listening for their ancient, cellular whisper. Then she would excuse herself politely and return to her office, where the piles of data and the silent microscope awaited her, unchanged by the world's applause. The Nobel ceremony in Stockholm that December took place under a sky of deep winter blues and early twilight. Barbara wore a simple, elegant dress, a corsage of pale flowers pinned to her shoulder, and sat among the other laureates in the great concert hall, its chandeliers spilling warm light over the solemn assembly. When it came her turn to receive the medal from the King of Sweden, she moved with a slow, deliberate grace. The applause rolled up to the high ceiling and seemed to hang there, a shimmer of sound. But the most telling moment, according to those who knew her, came later that night at the banquet, when she was asked what she would do now. She smiled slightly, her eyes distant and gentle, and said that she would go back to her laboratory and continue her work, because there was still much to learn. The statement was not grand. It was entirely in keeping with someone who had never studied plants for prizes, but for the quiet unfolding of truth. When she returned to Cold Spring Harbor after the celebrations, the winter landscape welcomed her with a familiar, deep silence. The cornfield lay under a sheet of fresh snow, the furrows smoothed into soft, white waves. The laboratory was warm, the same amber light falling across the same wooden benches. Barbara hung her coat on the hook by the door, sat down at her desk, and took up her notebook as if she had merely been away for a weekend. She continued to work for more than a decade after the Nobel, still growing her corn each summer, still peering through the microscope with undimmed curiosity. Younger scientists would come to seek her advice, and she would receive them with the same attentive stillness she once gave to her chromosomes. She had become, without seeking it, something of a sage, but her manner remained that of a dedicated gardener, one who knew that the best flowers grow without constant admiration. In those final years, her daily routines softened slightly. She took longer walks along the harbor, watching the sailboats tilt on the summer breeze, or sat quietly on her porch as the evening gathered. She often spoke of the feeling for the organism, a phrase that encapsulated her entire philosophy of science. Understanding, she believed, arose not from imposing theories on nature, but from a deep, empathetic attention to the living thing itself. This was the secret she had learned as a child lying in the grass, watching ants, and she had never abandoned it. Her life's work had been a single, sustained act of listening to the corn, to the chromosomes, to the slow, patient music of the natural world. And now, in the glow of her twilight, the world had finally quieted itself enough to hear what she had been saying all along. The jumping genes were not just a fact about maize. They were a revelation about life itself, a reminder that, even at the deepest, most fundamental level, existence is dynamic, creative, and forever in gentle motion. The final chapter of Barbara McClintock's life unfolded with the same gentle, unhurried tempo that had marked all the years before. Into her late 80s and early 90s, she continued to live in her cottage among the oaks, rising early, taking her simple breakfast on the porch, and walking the short gravel path to the laboratory. Her step had slowed, and she sometimes used a wooden cane, but her eyes were still bright and her mind still sharp as a freshly honed scalpel. The cornfields behind the lab continued to be planted each spring by assistants who carried forward her methods, and she would visit them as the summer heat built, standing at the edge of the rustling green, her hand resting lightly on the top of the cane. The harbor water, visible from her porch, took on different hues through the day: slate gray at dawn, deep blue at noon, liquid copper at sunset, and she became a quiet connoisseur of this daily show. In many ways, her life had come full circle, back to the solitary, observant stillness of her childhood, now enriched by decades of a deep, committed conversation with the living world. She received visitors, but only those who understood the value of quiet presence. A few times a month, a younger colleague would come to sit with her on the porch or in her small office. They would talk about new findings in genetics, about the startling discoveries in epigenetics that were extending the implications of her transposon work into entirely new realms. Barbara listened with intense focus, nodding slightly, occasionally interjecting a sharp, clarifying question. She never spoke with the possessiveness of a discoverer defending her territory. Instead, she was simply delighted to see how far the territory had expanded, how many new paths were being traced by younger explorers. When these visitors left, walking back down the gravel path under the canopy of trees, they often felt they had not merely been advised, but had been, in some quiet way, blessed. The changing of the seasons at Cold Spring Harbor remained the primary calendar of her heart. She was particularly fond of the autumn, when the air smelled of wood smoke and fallen leaves, and the migrating geese crossed the pale sky in honking V's. On such afternoons, she would sit for an hour on the wooden bench by the pond, her cane propped beside her, watching the shifting pattern of shadows on the water. The ducks, long accustomed to her presence, would paddle close without fear. It was a scene of profound peace, a landscape that had absorbed decades of human endeavor without losing its own identity. The laboratory buildings stood in the distance, their windows catching the low, golden light, and inside them, young scientists carried on the work that her patients had made possible. She felt no need to be among them. It was enough to know that the inquiry continued, that the listening had not stopped. As her physical strength waned, her inner world seemed to deepen rather than contract. She spent more time with her notebooks, not so much writing new observations as rereading old ones, tracing the long arc of her own thought. The pages were filled with her neat handwriting, the ink now faded to a warm sepia, alongside drawings of kernel patterns and chromosome structures. She could remember, with astonishing clarity, the day each observation was made, the quality of the light, the sound of the rain on the roof, the feel of the slide under her fingers. Her memory was a vast, well-tended garden in which she could walk at will. A few close friends suggested she dictate a memoir, but she waved the idea away gently. Her life, she felt, was already written in the maize plants themselves, in the gene maps, in the minds of the many researchers who now grasped, without struggling, the dynamic genome she had first glimpsed in solitude. Barbara McClintock died on September 2, 1992, at the age of 90, in the Huntington Hospital near her home. The day was reportedly mild and clear, the kind of early September day when the first hints of autumn yellow are just beginning to touch the leaves. She had never married, never had children, but in the largest sense, she had nurtured a vast intellectual progeny scattered across the globe. At her request, there was no elaborate funeral, no grand memorial service. She was cremated and her ashes returned to the earth she had so patiently studied. At Cold Spring Harbor, the cornfield was harvested that fall as usual, the golden ears gathered and catalogued, the stalks cut down and turned back into the soil to nourish the next planting. The great wheel of the agricultural year turned, as she would have wished, without ceremony, without interruption. Today, if you walk the grounds of the Cold Spring Harbor Laboratory, you can still feel her presence woven into the very texture of the place. The small white buildings still stand, dappled in the shade of the old oaks, and the corn still grows in a plot behind the lab, tended by researchers who understand that they are custodians of a legacy. At dusk, when the light softens and the harbor turns to a mirror of pale gold, it is easy to imagine her walking slowly toward the water, her hands loosely clasped behind her back, her gaze both inward and outward. The world of science has long since accepted her great discovery. The jumping genes have been found in every organism studied, from bacteria to humans, and they are recognized as powerful drivers of evolution and disease. But the deeper lesson of her life transcends genetics. It is the lesson of patience, of quietude, of the profound power of paying attention. In a noisy, hurried world, she showed that one could wait for the truth to emerge, as a seed waits under the soil for the right warmth and light. And in the end, the truth broke through, not with a shout, but with the slow, irresistible force of a living thing reaching for the sun.